Hello friends, welcome back to my channel and welcome to another video tutorial from Basic Biology. So today I am talking about the mode of replication. In our last video I told you that replication occurs in a semi-conservative fashion. That is, one strand is coming from parent and another strand is synthesized newly on the template strand. But how it has been proved? There is a beautiful experiment performed by two scientists, one is Meselson and another is Stahl, to explain that DNA replication occurred in a semi-conservative fashions. They got many evidences regarding it. So how they performed this experiment? In 1958, these two scientists grew E. coli in their lab within the medium containing the ammonium chloride. And this nitrogen containing ammonium chloride is specifically designed with the heavy isotope nitrogen. That means this NH4Cl with the 15N is used instead of the normal 14N nitrogen. So this heavy isotope is used to culture those bacteria. So as a result, all the bacterial content including the DNA are made up of these 15N nitrogen molecules. Now after successfully growing within this 15N medium, they transferred the E. coli bacteria into another medium that is called 14N medium, which is a normal nitrogenal medium. So that within the medium, after here also it's growing, after successfully culturing those bacteria, they analyze their DNA to know about the situation or the replication status. But one thing you have to know, that the separation of this 15N and 14N is possible due to a specific technique which is known as density gradient centrifugation. And in this technique a specific chemical is used which is known as cesium chloride. Now this cesium chloride is used to produce a linear concentration gradient to explain the banding pattern as well as to differentiate the 15N isotope or 15N and DNA or the contents from the 14N DNA or the 14N constituents. Now how it is possible? So let us see. After successfully growing and the DNA were extracted from the E. coli, and in the replication first cycle, it has been found that previously when the bacteria are present in the 15N media, the only band is that is N15 band. This is the heavy isotope band because the content of the solution is N15. So all the bacterial content including the DNA are leveled with the N15. So the DNA also leveled with the N15 and the band appeared only the N15 band, that is the pure band. But after successfully proceed into a replication process, that is the cycle 1, first replication or first generation of replication, then it's a very surprising result appeared, that is completely elimination of these N15 band, and there appear a new band which is known as N15N14 band, that is a hybrid band. So that is what is the peculiarity, that if the replication process occurred in a conservative fashion, that means the N15 band had to survive or had to exist in the next generations also. But here we see that there is no N15 band appeared in the first replication cycle. So it indicates that replication never ever occurred in a conservative fashion. If it occurs, then this band again exists in the next cycle, but it doesn't happen over here. Now There is again confusion that this N15 N14 band may appear in two cases, whether it is semi-conservative or whether it is dispersive, because in both cases the mixed band appeared. So that is what is the difference. So this N15 N14 does not indicate that it is semi-conservative. It is either semi-conservative or dispersive. So we move to the next replication cycle, where we again found another surprising result, that is a new band appeared, that is the N14 band, along with the N14 band. So along with these N14 band, another new band appeared, that is called N14 band. So it indicates if the product is the mixed product, so definitely this band exists. But here we found another new band N14. So it indicates that replication occurred in a semi-conservative fashion, because all those newly synthesized strands are labeled with the N14 isotopes. So here we see the diagram. So in case of the N15 N14 hybrid, the band is appeared over here, but when after the denaturation the new strands are like that, here the newly synthesized strand is labeled with the N14 and the hybrid band also exists. And if we see the next cycle again, then we can see these three bands are labeled with the N14 because they are newly prepared, and one hybrid band exists. So it has been confirmed that the replication occurred in a semi-conservative fashion. If it occurs in a dispersive fashion, then in all cases only one band exists, that is the N15 N14, but that never happened over here. Now one thing you have to note, that while the replication is growing on or replication is increasing, the pattern of banding is also changing, because most of the time questions appeared or questions are asked, that is, what will be the banding pattern after the fourth generation of the cycle or the fifth generation of the cycle. So here is the answer. If we see the diagram very carefully, then one thing is very clear, that the N14 band with the growing cycle of replication become thicker. That N14 band become thicker, and N15 band become thinner. N14 band become thinner. So in this way, Meselson and Stahl proved that replication process always occurred in a semi-conservative mode, rather conservative or dispersive. If you have any queries, then definitely you can ask me or mail your questions. In our later video, I will be talking about the replication in prokaryotes. So stay tuned and follow my channel. Thank you.